Hello viewers, welcome to Mesology. In this session, I will focus on the internal anatomy of the human kidney. Human kidney are the major excretory organs. The unit structures in the human kidney are called uriniferous tubules. They can also be called by the name of a nephron, but better to remember it as uriniferous tubules because you can then connect to the formation of the urine, which is its function. And that becomes easy to remember both the ways. Now, in each kidney, there are around 1 million to 1.2 million uriniferous tubules. And these uriniferous tubules range from 4 to 5 centimeter in length, or we can also say it can vary from 30 millimeter to 55 millimeter in certain cases. Each uriniferous tubule comprises of the following parts. It has a cup-like closed end which is called the Bowman's capsule, and this Bowman's capsule has a inner lining and an outer lining of cell, and then the outer lining of the cell then pulls out into a next tubule which is much convoluted in shape, and that's called the proximal convoluted tubule. Why proximal convoluted tubule? Because the word proximal means close to or nearer to, and who is it nearer to? It's nearer to the Bowman's capsule. So the justification of the name PCT is indeed because it is the closest tubule to the Bowman's capsule. What happens to the other side of the PCT? It draws down into a much thinner U-tube-like structure. And this U-tube-like structure was named by a scientist, and that's why we call it as loop of Henle after the name of the discoverer. This loop of Henle has a descending part, and then it goes into an ascending part, and that ascending part is little away from the Bowman's capsule. And at the end of the ascending part, it again has a convoluted structure, and that convoluted tubule on the ascending part of the loop of Henle is called the distal convoluted tubule. Again, why distal? I hope you can indeed give a justification, because the word distal means distances away or away from. Now, it is away from whom? It is again away from the Bowman's capsule as compared to the proximal convoluted tubule. Now, this distal convoluted tubule further runs out into a collecting duct. Is that all the structures who are involved in this excretory system? Definitely no, because the blood has to flow through these tubules. Only then the blood can be filtered, and that can be extracted of the unwanted substances, mostly excess of water, the nitrogenous waste, excess of salts, and any toxic substances. For this, who brings the blood into the uriniferous tubules? When it concerns to the kidney, the renal artery brings the blood to the kidney. And now when it has to enter into the uriniferous tubule, since each kidney has 1 million of uriniferous tubules, so this branches out into million of the arterioles, and these arteriole, one in each of the uriniferous tubule, take an entry with the name of afferent arteriole, and that goes and fits into the cavity of the Bowman's capsule. And when it fits into the cavity of the Bowman's capsule, it becomes a network of capillaries. A network of capillaries fitting into the cavity of the Bowman's capsule. And that is the structure where the blood comes with force and it builds up a very high hydrostatic pressure for ultrafiltration. Further, from the other end of the arteriole, where it emerges out in the form of efferent arteriole. Why do we not call it by the name of afferent arteriole again? Because the entering arteriole had a much broader diameter compared to the exiting arteriole, which has a much thinner diameter. And this assists in the process of ultrafiltration largely. So we have two different names, the entering arteriole as the afferent one and the exiting arteriole out of the Bowman's capsule as the efferent arteriole. Then what happens to the efferent arteriole after it would have come out of the Bowman's capsule? It now winds around the renal tubule, right from the proximal to the loop of Henle and then further around the distal convoluted tubule. These are called the peritubular capillaries. They also assist in the functional activity for the formation of urine, and those go by the names of tubular reabsorption and tubular secretion. In the next session, I will be explaining the function or the formation of the urine under the three heads. But here this peritubular capillaries, when it emerges out from the distal convoluted tubule, each of those peritubular capillaries from one and all of the uriniferous tubule will reunite and form the renal venule and then finally the renal vein. Now they will be taking the blood back to the heart because the blood has been filtered off the waste products and it needs to return to the heart for oxygenation once again. Now coming to the part of the cortex and the medulla. I spoke to you, the cortex looks darker and the medulla looks lighter in a section. But why? Into the cortex fits the Bowman's capsule, the glomerulus, the PCT and the DCT. PCT for the proximal convoluted tubule and DCT for the distal convoluted tubule. Then what are we left with? We are just left with the thinner parts, that is the loop of Henle, and then we have a tubular part which is called the collecting duct. Only these two structures are seated into the medullary region, which is the innermost part of the kidney. But these thin thread-like structures, how will they be placed so safely without falling into knots? That is also to be seen. For this we have extensions of the renal cortex through the medulla, and these are called the columns of Bertini. The columns of Bertini spread out into the medulla between the pyramidal cells and ensure that the thin tubular parts, that is the collecting duct and the collecting tubule and the loop of Henle do not form into a knot and do not prevent the proper filtration of the blood passing through it. We have another duct, we call it as the duct of Bellini. Now what will this duct of Bellini do? When the collecting ducts are coming out of the pelvic region of the medulla, the pelvis is indeed a little swollen part of the ureter. emerging out of the kidney. And here, this is the ureter, which is actually formed by the union of large number of collecting ducts that is tightened before it comes out of this region by the ducts of Bellini, so that the safety and security with which the ureter would be formed would have no overlapping of the collecting ducts in them, and thus ensure a proper filtration and a proper excretion through the excretory system. Thank you for watching. Please do like, share and subscribe to my channel. Thank you once again.